My name is Zachary Stansell. I am the National Plant Germplasm System Hemp Germplasm Curator. We're based in Geneva, New York, and my role is to collect, conserve, characterize, evaluate, and distribute hemp genetic resources to an international hemp research community. So what we're looking at today is part of our germplasm collection. So we're growing over 500 unique accessions in this field, and we're measuring nearly every kind of trait you can imagine. So the goal is to have a big, genetically and geographically diverse hemp germplasm collection, and to link rich phenotypic data to those accessions, as well as capture large amounts of genetic data. And so this can enable breeders and plant researchers to more quickly and efficiently understand these priority agricultural traits in hemp, as well as breed locally adapted, climate change resilient varieties. One of the main goals of this work is to shave years off of that breeding process. So breeding a cultivar can take 5, 6, 7, 8, 10 or more years. And a lot of that early stage is looking at a lot of diverse germplasm and measuring its phenotype for your trait of interest, right? So what if we can help breeders, A, get access to diverse germplasm, but B, very quickly identify a high or low value for a trait that they're interested in. So for example, maybe a breeder is looking to grow something tall in Florida. They can go to GRIN Global and search our database for plant height. Or conversely, they can look for plants that flower late, which is likely to be important if you're growing hemp in Florida, for example. We distribute seeds from these accessions to researchers and breeders all over the world.
This is the USDA-ARS hemp collection, and we have accessions replicated out in this plot where we're taking data on fiber and grain quality, plant architectural traits, and secondary metabolites. And we have collaborators throughout the country. Some of this work is being replicated in Oregon, Washington, Alabama, and Louisiana. We built these are called hemp huts, and what they are is the bottom 3 feet is polycarbonate, and then the top 3.5 feet is 15 micron mesh, so it's breathable fabric. And we put this polycarbonate on the bottom so as the mowers go through it doesn't puncture the mesh, and then the mesh is breathable so it can let air pass through and water pass through, so these, they can breathe, I guess, yeah. And it also prevents pollen from getting through.
We are working with a close collaborator to genotype this collection as well. So there's about 2,000 individual plants from this field that we've collected tissue from that will generate low pass, long read sequencing data for and make that publicly available.
We're working with Breeding Insights to come up with markers for around 3,000 loci to help identify the traits in all of our hemp collections. Specifically, I took tissue from every plant in the field, and we are going to run several tests to identify those markers and try to locate terpene synthases.
The U.S. government had a program, Hemp for Victory, where they encourage growing hemp throughout the Midwest. And it's fiber hemp, so it's these tall, narrow varieties with not as many stems to convert the phloem tissues to fiber, and that fiber could then be used for ropes during World War II. And then after World War II, those programs were shut down. Hemp production was grown at a limited capacity, and you had these escapes from the fields, and those escaped varieties then started growing in ditch banks as ditchweed. Those remnants can still be found today.
Here is a feral variety. This was collected by Jeff Karstens in Iowa. And here we are looking at seed shatter. So this bag is about ready to be harvested. And we'll measure the ratio of seeds in the bag versus seeds on the inflorescence as a sort of a preliminary assay of shattering. The seeds in these feral types shatter readily. So they're just falling.
So we bag these just a little bit after flowering. This is a 360 micron mesh. And then we zip tie these on and just leave them till they senesce. And then we pull those bags down. We're dropping those out onto a weigh pen. And so how much shatters out, and then we thresh those. Then we thresh whatever's left. So we get like a shattering weight and a not shattering weight. Then we know so what's the percent that shattered.
So there's over 4,000 individual plants that we've been phenotyping on a weekly basis. We measure everything from the sex of the plant. So cannabis is a dioecious species, which means you can have individuals with male flowers and individuals with female flowers. But you can also have monoecious individuals, so individuals that have both male and female flowers. Some of the earliest accessions flowered in June, and some of the latest accessions haven't even started to flower yet, which is mind-bending because we're well into October. So when hemp starts to flower, generally it doesn't grow a lot more. So from a biomass or a fiber grain production perspective, it's good to flower late. If you're grown in Southeast Asia, you can take a lot of time, generate a lot of biomass, and wait longer to flower and make more seeds. If you're in Siberia, you have maybe 100 frost-free days per year. So it makes a lot of sense to flower really quickly and make your seeds and complete your life cycle. A lot of the cultivars that were bred for Northern Europe flower too soon in New York. We're surprisingly farther south than a lot of these Northern European production locations. So this is evidence that these plants have adapted to some very different environments. So other traits that we measure are things like plant architecture. So how tall does the plant get? How wide does the plant get? What's the length of the trunk? Things of this nature have really big consequences for downstream agronomic production. Some of these guys in here are over 12, 13, 14. I think there's some 16-foot individuals in here. We're also collecting floral tissue. So this plant right here is about ready to take a chemical harvest. So with our collaborators in the USDA, we will analyze about 20 different cannabinoids. So the famous ones, but also obscure boutique cannabinoids that may have a lot of medical value. So we're looking at all of this data, and we will upload this data to GRIN Global and link it to its accession. And here we are looking at seeds. We'll send an aliquot of these seeds to a different USDA-ARS collaborator who will measure fatty acid and protein content of these seeds. So we know that there is some genetic variation in the omega-3 fatty acids in hemp. So we're trying to look at all of this diverse germplasm and identify any accessions with unique fatty acid profiles. We're also interested in measuring this idea of fiber quality. What we'll do is we'll chop out these stems from the field and then send them to another USDA lab that has been working really hard to measure and understand hemp fiber quality. Traits of interest include things like how strong are the fibers? What's the fiber diameter? What's the fiber fineness? How does fiber fineness change along the length of the fiber? There's just a lot of questions we don't know about hemp. It's an old, new crop. And it's really fun to work with all these different smart people that are figuring it out. When I'm doing my job correctly, I am supporting many other researchers who are working on many interesting and complicated and confusing questions in hemp. So I really like that. It's fun to learn about all of the incredible research projects that are going on, and it's an honor to play a part in all of this really important work.